Hello everyone, let's get started. Today we're going to be talking about vaccination. A vaccine is defined as a substance that is used to stimulate the production of antibodies and provide immunity. Vaccines can be produced to protect against viruses, and vaccines can be used to protect against bacteria. One of the first questions we need to ask ourselves about vaccination is why? Why do we bother vaccinating people? There are really three answers. First, we want to protect individuals against contracting that particular infectious disease. And certainly, vaccination, first and foremost, protects the individual who receives the vaccine. Second, though, is that vaccinations promote what we call herd immunity. This is a term that we use in public health to describe the positive effects that vaccination has for an entire community. We'll talk a little bit more about herd immunity in just a minute. The third reason that we vaccinate is one you may not be familiar with, and that's to prevent certain types of cancer. We'll talk just a little about that in a minute as well. You should know that we have been vaccinating animals and human beings for quite a long time now. In fact, Edward Jenner developed the first version of the smallpox vaccine way back at the end of the 18th century in 1796. So vaccines are not new to medicine. So let's take a minute and talk about this concept of herd immunity. Herd immunity, again, looks at the entire population of people and how best to protect the population from an infectious disease instead of focusing down on the level of the individual. It turns out that if we can vaccinate between 60 and 90% of a particular population, we can achieve this goal of herd immunity. This concept was first developed in veterinary medicine when it was discovered that if we could, again, achieve 60 to 90% immunity in a herd of, say, cattle, that vaccination rate or that immunity rate would protect all of the herd from this particular infectious disease. Now, how does that work? Well, it turns out that if you can achieve 60 to 90% immunity, either through vaccination or through natural infection, you're going to achieve three results. And these look similar to the reasons that we vaccinate. Herd immunity will protect individuals, yes. But herd immunity will also reduce the prevalence of that pathogen in the environment. It will literally reduce the amounts of that pathogenic microbe in the environment. And third, and perhaps most importantly, herd immunity will protect vulnerable members of a herd or a population. And by vulnerable, I mean people who either can't be vaccinated or can't respond to a vaccine. Who are these vulnerable people? Well, very young infants are a good example of that. Very young infants will not respond to vaccination. Elderly people who have reached an age where their immune system is not working as well as it once did need protection. People who are chronically ill, who are fighting diseases on a chronic everyday basis, also have an issue with their immune system not being quite as strong as we would hope it would be. And finally, there are people in any population who cannot be vaccinated, either because they are allergic to something in the vaccine or because they've had an adverse reaction to a vaccine in the past. These are the folks that we're trying to protect when we vaccinate 60 to 90% of a population. Let's take a minute and talk about this very interesting idea that certain cancers can be prevented with vaccination. Probably the most commonly used cancer prevention vaccine is a vaccine against the virus called human papillomavirus, or HPV. HPV is a sexually transmitted virus, and HPV infection can lead to the development of cervical cancer in women. In fact, it's been shown that 90% of all cervical cancers can be traced to HPV infection. You may not have been familiar with the fact that certain viruses can trigger cancers, but HPV is one of those viruses. Another virus that can trigger cancer is hepatitis B. So by vaccinating people against HPV and against hepatitis B, we can reduce the prevalence of cervical cancer and liver cancer that develops as a result of those types of infections. Now, one thing I want to mention just briefly is that it is possible right now, in a couple of limited instances, to actually treat cancer with vaccines. Now, these are not the vaccines that we've been talking about so far. These are not vaccines that are directed against a pathogen per se. These are vaccines that are directed against a cancer instead. A good example of this is a product called Provenge, which is on the market as a treatment for advanced prostate cancer. You're looking at the label from the Provenge product over here, and also what this product looks like in terms of the infusion equipment. So if you look at the label, and I know it's hard to read, but it says that this product is going to have a minimum of 50 million autologous, essentially T cells that have been activated with cells suspended in lactated ringers. Let me explain what this means. The physician essentially removes some of the patient's immune cells and some of their cancer cells and takes those cells into the laboratory and allows them to interact with each other. In other words, we're going to bring some of the patient's immune cells in direct contact with the cells of that advanced prostate cancer. And we're going to allow the cells to recognize the antigens on that cancer and start doing what they do, which is become sensitized to the cancer, present the cancer to other immune cells, and develop antibodies. Once that interaction takes place, those cells can be put back into the patient's body, and they will then work to fight that cancer. One of the beliefs that has emerged about treatment for cancer is that we need to pay more attention to helping the person's immune system fight the cancer, because the immune system is best set up to fight a cancer. In other words, if we could somehow assist the immune system in recognizing the cancer, the immune system will do a better job than any drug we could possibly give. at getting rid of that cancer for that patient. So Provenge is an example of a truly individualized approach to cancer therapy. The next question we need to ask ourselves is who gets vaccinated in the population? Obviously, infants and young children are routinely vaccinated against some of the most common infectious pathogens. Everybody knows that young children get a series of vaccines before they go, for example, to public school. It's also true that teenagers and young adults, college-age adults, are also vaccinated. There are certain adults that are routinely vaccinated as well. Pregnant women, for example, when they come in for their routine prenatal care, will be evaluated to find out which vaccines they've had, which ones they might need, which ones might need to be boosted, and so on. The idea is that we want the pregnant woman to be fully vaccinated as we can get her, so that she will produce antibodies that she can then pass on to her infant when it's born. The elderly, again, are a particularly vulnerable population. They are more susceptible to certain infectious diseases. And so there are some vaccines that are particularly given to elderly adults to help protect them against these infectious diseases. There are several at-risk groups of individuals who, because of the work that they do, get exposed to some pathogens that other people in the population don't get exposed to. For example, members of the military are sometimes vaccinated against anthrax because anthrax is a potential bioweapon. Remember, the organism that causes anthrax, Bacillus anthracis, is one of the types of pathogens that is capable of sporulating. And for a while now, we've been concerned that a foreign enemy might use anthrax spores as a bioweapon. So we want to protect our military members from possible exposure to Bacillus anthracis. Various healthcare professionals get exposed to pathogens that, again, the general public doesn't come in routine contact with, and they will be fully vaccinated according to the risks that they face. Veterinarians are exposed to certain pathogens. A good example of that is the rabies virus. Veterinarians are routinely vaccinated for protection against rabies, since they come in contact with animals that might be carrying that pathogen. Last on the list is travelers. Now, people who travel don't necessarily need any particular vaccines, but people who travel to certain parts of the world where unusual infectious pathogens are still around are routinely vaccinated against those. For example, if a traveler is going to go to a part of the world where yellow fever is still a problem, that traveler will be advised to get a yellow fever vaccine. Let's take a minute and think about what vaccines are actually made out of. Vaccines all have to contain some form of the causative agent that we're trying to stimulate an immune response to. So all vaccines have either all of the cell or particle from a pathogen or some piece or part of a pathogen. They have the causative agent material inside. And the reason, of course, that that's important is that it's that material that will contain the antigens that the immune system will respond to. So all vaccines have some form of the causative agent, the infectious pathogen, inside of them. Now, in the old days, vaccines were primarily prepared as whole cell preparations. Now, again, most vaccines are directed against viruses, and viruses are not cellular, but the concept is the same. The concept is that you take the entire viral particle and you put it into the vaccine and you allow the immune system to respond to it. When vaccines are directed against bacteria, again, in the early days of vaccine development, the entire cell would be used in the vaccine. What we now understand is that you don't need to put an entire viral particle or an entire bacterial cell into a vaccine for it to work. And in fact, you can get better and what we would call cleaner immune responses to only parts of the viral particle or parts of the bacterial cell. The parts that are the actual antigens, the actual thing that the immune system is responding to. Now, there is some language around vaccines that can be very confusing to people. We often refer to vaccines as either being killed or attenuated. Another term you'll hear for attenuated is modified live. One of the greatest fears that people have about vaccines is that somehow the vaccine will give them the disease that it is supposed to protect against. And you can understand where that fear comes from. If you tell a person that there is viral particles or there is bacterial material inside of a vaccine, you can understand why they would fear getting the disease from the vaccine. In reality, vaccines cannot cause disease because the material that is inside the vaccine has been altered so that it is no longer able to cause that infectious disease. So when a vaccine is labeled as killed, what that means is that the material, the causative agent material that's in the vaccine has been destroyed. In comparison, when a vaccine is labeled as attenuated or modified live, what that means is that the cells or particles have been modified in the laboratory so that they're no longer able to cause infection, but they have not been destroyed. So there are two types, two big categories of vaccines in how the causative agent is present in the vaccine, whether or not it's been destroyed or whether it's been modified so that it is present but it cannot cause disease. Either way, whether you receive a killed vaccine or an attenuated vaccine, the vaccine cannot cause the infectious disease because there is no infectious material in a vaccine. Now, the second material that is commonly present in a vaccine is a preservative. And preservatives have received some attention in recent years because we have used preservatives in the past that are toxic. And while the amounts of preservative that were put in vaccines did not necessarily cause harm, we now recognize those materials as being toxic, and so we no longer use them. You may have heard, for example, that we used to use a preservative called thimerosal, which is a derivative of mercury. It's a great preservative, but of course, mercury is a terrible toxin, And so we no longer use thimerosal in vaccines. We use primarily more natural preservatives now, including things like vitamin E products. The reason that we put preservatives into vaccines is to increase the shelf life of the vaccine. When we produce vaccines, we have to think about how easy it is to give that vaccine to large populations. And one of the things we have to consider is shelf life. It doesn't make a lot of sense to produce a vaccine that has a very short shelf life. It's unlikely that that vaccine will be very practical for use. Now, there are two other things on this list of ingredients. One is called diluent, and one is called adjuvant. And you'll see that I have plus minus symbols next to each one of those, because these are not necessarily found in every vaccine, but are found in certain vaccines and certain formulations of vaccines. All a diluent is a fluid, usually a saline-based fluid, that gets added to a dried version of a vaccine in order to dilute it so that it is in an injectable form. If you look over here on the right side of the slide, you can see a vaccine that has been produced commercially that comes in two vials. There's one vial that contains a freeze-dried preparation of the vaccine, and there's another vial that contains what's labeled as sterile diluent. At the point of delivery, in the doctor's office, or in the public health facility, or in the field, wherever it is that the vaccine is being delivered, the person who is administering it draws up the diluent from this vial and places it into this vial so that the dried vaccine material is resuspended into the liquid. Then that can be drawn up and given to the patient. So diluents are also there to help vaccines be more practical and to have a longer shelf life. When vaccine material is in a freeze-dried formulation, it just lasts longer. Once the diluent has been added, once the vaccine is in solution, in other words, the clock is ticking on how long that vaccine is useful. So we wait until the minute that we're about to administer the vaccine before we dilute it. So again, it's a measure of practicality that we can add for vaccines when we use a diluent system. Last on our list is an adjuvant, and you may or may not have heard this word before. An adjuvant is a material that you can add into a vaccine, again, not all vaccines, but certain vaccines, when you want to increase the stimulatory effect of the vaccine. There are some vaccines that contain antigens that just aren't that good. Remember, we said that different materials have different amounts of antigenic stimulation. The largest proteins make the best antigens. Things like DNA and RNA make very poor antigens. So depending on the causative agent material that's inside a vaccine, the antigens may be very strong or they may not be very strong. So if there's a vaccine that has poor antigenic potential, it makes sense to add an adjuvant to that vaccine, to add a chemical that we know will stimulate a strong immune reaction.